Hello all, today we'll discuss about the anatomy of limbus. So limbus is the junction between cornea and sclera or the junction between the cornea and conjunctiva. Left hand side, that square box, it is shown black color, that is the corneal limbus. Okay, or the above diagram you can see that red dotted lines, circular dotted lines, that is the limbal area. So the bulbar conjunctiva is a conjunctiva which we can see, okay, when my eyes are open. You can see it is denoted by the purple color, and palpebral conjunctiva is the bluish one that is within our eyelid. And limbus is a junction between the cornea and sclera or the cornea and conjunctiva. Anatomically, the limbus refers to a circumcorneal transitional zone of the conjunctivocorneal and corneoscleral junction. Circumcorneal transitional zone of the conjunctivocorneal and corneoscleral junction. So the XYZ hypothesis states that it is proposed on the basis of experimental evidence that both limbal basal and corneal basal cells are the source of corneal epithelial cells. The corneal epithelium is maintained by a balance among sloughing of cells from the corneal surface is equal to cell division in the basal cell layer and plus migration of the basal cells originating from the limbal stem cells. So here you can see the limbal epithelium. You can see the limbal stem cells, right? And you can see how this limbal stem cells is going to the corneal epithelium. And this, beside the limbal stem cells, those are transient amplifying cells, okay, which gives rise to the corneal epithelium cells. Here also you can see these are the cells. You can see the red one is the blood vessels, then comes the terminally differentiated cells, okay, then this one is the basement membrane, then the transient amplifying cells, this brown ones are the melanocytes, then the violet ones are the limbal fibroblast, and then comes the limbal epithelial stem cells. Okay, so these are the cells of the limbus basically, which is present at this junction. Anatomical limbus, conjunctivocorneal junction. So what is that? At this point the bulbar conjunctiva is firmly adherent to the underlying structures. Okay, the substantia propria of the conjunctiva stops here, but its epithelium continues with that of the cornea. At the transition zone, the epithelium becomes several layers thick, right? That is 10 to 12 micron and arranged irregularly at the limbus. So this is all about the conjunctivocorneal junction. Now coming to the sclerocorneal junction. At this point, the transparent corneal lamellae become continuous with the oblique, circular and opaque fibers of the sclera. So conjunctivocorneal junction is nothing but the cornea and conjunctiva connection, right? So how the structures over there, that you need to know. So here in this diagram you can see the corneoscleral limbus. You can see the demarcation of the corneoscleral limbus. Okay, the haze part is the sclera, okay, and you can see the corneal scleral limbus nicely over here. So the same thing it is showing, okay, the site where the conjunctiva becomes closely attached with the fascia bulbi and the sclera. You can see the bulbar limbus, you can see the corneal limbus, and you can see the conjunctival limbus region where the site where the conjunctiva fuses with the cornea. So these are the sites what you should demarcate while demarcating limbus. At limbus, the corneal epithelium becomes bulbar conjunctival epithelium. You should remember that. The Bowman's membrane becomes continuous with the lamina propria of the conjunctiva and Tennant's capsule. Stroma becomes sclera at limbus. Okay. The Decimex membrane becomes Swalb's line at limbus. Endothelium lines the trabecular meshwork and becomes continuous with the anterior surface of the epithelium. So these are the very very important points which you must know what is happening in the limbal zone. Thank you.